Audio is recording. Let's see, audio check, and the audio is recording. Okay, let's go ahead and get started. My name is Robert Mazzeo, and I will be your professor and instructor for cell biology, or PCB 3023. So let me start by saying, if you registered for this course under the name Robert Hill, or with the professor being Robert Hill, that's the same person. So I hope that didn't cause any confusion to anyone. I just had to make a change with the college. So okay. So this is cell biology. Cell biology is an amazing course. It's one of my favorite courses, if not my most favorite course. And my hopes is, if you really excel at this course, you do really well, at the end of the course, you will see the cell as a very complex machine, something that's very manageable in terms of, can I understand it? And being able to see how the proteins are made, how they're trafficked from organelle to organelle, how all the different organelles function together, and how the cell really brings those functions together. It really paints a pretty good story of the cell, and I think that's and that's my goal for you in this course, is at the end of it you really start to see the story of the cell. So let's do a couple things here. First, I want to share my screen so we can get the syllabus up. And that would be right there. Correct? Okay. All right. So just a couple words, just preliminary words, just to get this out of the way about the course. This course is very challenging. I'm pretty sure that this is going to be one of the more challenging courses that you take in your college career. And what makes it challenging is really the complexity of the subject. That's simple. Imagine having to learn a new language, right? And then in that language, learn how an internal combustion engine functions in extreme detail. And that's similar to what you're going to do in this course. The trick at first is going to be learning the vocabulary, getting up to speed on that. So if you did really well in Bio 1 cellular processes, that's a great thing. That is going to give you a really good background for the information that we do in this course. What you're going to see is that really over the first 1 or 2 lectures, I pretty much give you a review of probably about 60-70% of that whole course. And then from there on, we're going to be learning a lot of new concepts. So this course is fully online. It is offered 2 times a year. So we offer it in the fall and the spring. I teach it both semesters. In the fall, I do teach it in person. So after this welcome video, when I go over the course, if you think that taking this in person would be better for you, I can completely understand that. This is the first time this course has been offered fully asynchronously online at St. Pete College. And I made that decision because I think it's something that can definitely be done. The one caveat to that, so, if you've taken a lot of online courses, my suspicion is that those online courses had a lot of electronic resources for the instructor to provide for the students, right? So if you took, probably Bio 1, I know Bio 1, Bio 2 has those. And what I mean by that are, exercises to help you get through the reading material, lots of different quizzes and interactive exercises that you can do. Unfortunately, this course does not have any of that, right? So there are no click your way to success exercises that I'm able to give you. This course is going to be lecture, study, and take challenging exams. That is basically the gist of it. I also have some, research like exercises that I've worked in to kind of, help boost your grade a little bit. But, for the majority of this, what you're going to be doing is, reading the text, watching the recorded videos that I have, and studying. I'm here to help you and succeed in this course. But success in this course, I can tell you, is going to take a tremendous amount of discipline on by yourself, right? You're going to have to stay up on the material. You're going to have to study it a little bit, preferably every day. And that's because that's really the way that you learn the language. You learn the vocabulary. You have to do it every day. Trying to get, down the material for these exams a day or two before, I think honestly that's going to be very, very well. In the past, I've seen that it's very, very unlikely to be able to do something like that. Okay. So the mechanics of this course, though, is really simple. You basically have exams, you have quizzes, and you have some data sets where you're going to analyze data. Okay. All right. So the best way to get in touch with me is Canvas messaging right there. Inbox, or however you guys do it. It's probably a little different on your end, but that is the best way to do it because I'm teaching other online courses this semester. And when you use Canvas messaging, I will know what course the person is contacting me from. You can also contact me via email. And then also I will see you guys, most of you at least, I believe it's every Thursday, right? Double check that here. Yeah, Thursday. Yep. Yeah, Thursday we have lab. Okay. Now, the lab is a separate course, right? You get a separate grade for the course. I don't make that component. The lab is not as difficult as the lecture. In all honesty, I really care more about the lecture than I do the lab, but that doesn't mean you can't get away with not doing the lab. But it is a completely separate course for which you get a completely separate grade. There's going to be significant overlap in content between the two, but I really wouldn't, there are a lot of things that we do in the lab that will reinforce what we cover in lecture and will help you learn it. But I wouldn't look at the lab as a tool to help you exceed in this course. All right. You're going to exceed in this course by understanding the lectures, by reading the book, understanding the book, working the problems at the end of the chapter. That's how you're going to succeed in this course. Okay. Contact me via Canvas messaging. So what I'm going to do every Monday from 11:30, I will be in my office. I will be on the computer. If you guys have questions, you just join the Teams meeting and I'm there. If I'm not available, that means I'm working with another student and you'll just have to wait. But it's just like open office hours. It's not a structured review session or anything like that. Okay, so come with specific questions. But you can also email me, message me, and set up a time to meet at other times. You're not bound by these hours at all. It's just hours I arbitrarily picked. And I have a very open door policy with you guys, okay? Okay. The book. You need the book. All right. And also, you need the most current version of the text because I can't vouch for the previous versions. In my experience, is the material the same? Yes, but it's all jumbled up. They will move stuff around, and that's going to create a lot of confusion to you in what you're responsible for the exams. So I know the book is a little pricey. It is in Barnes and Noble's first day. So I believe all you need to do is stay opted in, and you will receive a digital copy of the textbook. If you need a written copy, you can purchase that through the publisher. I don't think it's too much more. It's like a 3-hole punch copy. But a little bit about this book. There's basically two main textbooks that are used to teach this course at colleges. When I took it, I used the Alberts textbook, which is, if you take me from molecular biology, we use the Alberts textbook for molecular biology. And that was really the first, it was the gold standard for cell biology textbooks. And this guy, Mr. Cooper and Kay Adams. So Cooper is actually a science fiction writer and also a molecular biologist. And I notice his writing style is a little easier to kind of get into and follow along with in the text. And this is some pretty nerdy material, guys. I'm not going to lie to you. It can be very challenging to read. It's very dense reading. But I think, his approach to how he's written it has made it a little better, a little easier to swallow than the Alberts book. So I've enjoyed using this textbook, but unfortunately, it just comes with 0 online resources. There's no digital exercises that I can provide for you guys. Neither does the Alberts textbook. All right, so this is in first day access, so just stay opted in, and you will be able to get access to a digital copy of the textbook. Okay, so you have unit exams, and this is, look at that, 80%, right? Success in this course is really going to be contingent on your ability to do well in these exams. I will tell you a little bit about my teaching philosophy in this course and also genetics. It is very easy to get a C in this class, okay? But you really have to work for the B or the A, right? So keep that in mind going forward. If you're just looking to pass, get out of here, I can completely understand that, and you're going to have to take the exams and perform reasonably well on them, of course. But I do end up applying somewhat of a curve usually to most exams that kind of compensate for that. And but if you're a student that needs an A or B in here, you're going to work for it. Okay. So you have a fair amount of time to take these 40-question exams, but my questions are very, I consider them pretty complex questions, right? There's a lot of, 2 of the above, none of the above sorts of questions that are able to incorporate a lot of different things, a lot of different components of what we've covered in lecture. Okay. These are proctored exams, right? They are using the Respondus and HonorLock proctoring stuff. And, just a word on that. So, my work has pretty much been done in this course. I've got all the lectures are banked and recorded for you guys. I've already made up the quizzes. I've already made up the exams. I'm going to have to be grading these data set discussions as we go through them. But the point is why I'm saying that is a majority of my time is going to be spent reviewing, as exciting as it is, but as very necessary, is reviewing the video recordings of you taking these exams. So, if you're taking this course with the thought that maybe you can, use a little chat GPT while you're taking it and things like that, I'm going to be looking at these videos and the audio very, very carefully, at what you're doing on the screen, how quickly you're taking the exam. And I have some policies in place about this. So if there's suspicion that you guys, that one of you may not be taking the exam honestly, I'm going to require that you take it at a testing center, right? Or that, if you're at SPC, that you come in and take it at the SPC testing center. So just keep that in mind. SPC students are great. I've really never had, I've had a little bit of an issue with it, but not too much of it. So, I think that's all that needs to be said. Okay. All right. So now, just a little bit about these exams. I'm going to move over to modules real quick. So if you look over here on February 1st, there is a practice exam. Okay. This exam does not count towards your grade. What I would like you to do for this exam is listen to lecture 1. Okay. These are the chapter sections that are covered in that lecture. Read through the textbook, all right? And then by February 1st, which is pretty soon, all right? You're not given an enormous amount of time to study for this because it's not graded, right? It doesn't. It will be graded, but it doesn't work towards your final grade. But prepare for this because it will kind of give you an idea, help you gauge, are you studying enough for this course? The questions on this practice exam are fairly representative of the types and formats of questions that I will have on future exams. Okay, so, just so there's no surprises when you have exam 1, you'll get an idea of what sort of level of difficulty, you're going to be expected to know this information at, right? So I'm really expecting that you have this information mastered. And another reason I have that is because there are no dropped grades, right? You have 4 units, you have 4 exams, and none of those are dropped. The information in this class really builds on itself. All right. So if you have trouble, grasping the concepts in Unit 1, those troubles are going to be revisited when we have new concepts later on that require mastery of that content that we went over before. Okay. 40 questions, multiple choice, digital proctor. And you have, so this should be, let's check something real quick. I wonder if I messed this up. Yeah, okay. So you have an opportunity, if you haven't used Respondus and HonorLock before, all right, due January 23rd. At 5 p.m., okay, you have a little survey that I want you to take. And this survey will require use of Respondus and HonorLock. All right, the questions are, what's your name? Have you taken Bio 1? How did you do in Bio 1? Things like that. Okay, it takes 5 minutes to do. But use this as an opportunity to make sure that your settings are good, that you are comfortable, using Respondus. Instructions on how to set everything up will be given as you click through and work with that. You need to be working in a very well-lit setting. The camera needs to be centered on you. You're going to present your photo ID before you take the exam. It needs to be a very quiet, distraction-free environment. And you're basically, your eyes are going to be going from screen, right, to keyboard to screen to keyboard, and that's it. Like it shouldn't be really, and really you don't even need the keyboard. It should be just screen and using your mouse to answer the questions, right? So Respondus does look at that. It looks at your eye contact. Okay, so for these recorded lectures, you also have the PowerPoint slides, which are provided for you. Okay, you're looking at, each of these lectures has on average about 75 PowerPoint slides, 70 or 80 PowerPoint slides per lecture. A number of sections are covered. And okay, that's it. All right, I'm going to jump ahead. I'll get to the data discussions in a little bit. You also have a quiz, right? You have a quiz. Check one more here. Okay, so if you notice, the quizzes are not proctored, right? So the quizzes are up to you. If you want to, they are for a grade. So my recommendation is to, try to, do them closed book and then go back and check your answers before you submit them. But, the purpose of these quizzes really is just to boost your grades up. The quiz questions are not at all representative of the information that the types of questions that are given in the exams. Right? They're really easy. Right? These quiz questions are, just straightforward questions. You know, how many membranes are in the mitochondria, something like that. Okay. You know, you get these more complex questions on the exam. Okay. All right. But for each of those, you do have a quiz. If you'll notice, and this is one thing that gets students, but I have my reasons for not doing this. There are no review sheets. I don't do review sheets, not for this course, not for genetics either. And that's because I want you to learn the material, not just the material on the view sheet. All right, I get it. I understand what those do. But for this course, I don't do review sheets. Okay. All right. So you have those lecture slides. I also want to talk a little bit about the tutoring resources. And I think what the gentleman that does the tutoring for you guys, his name is Zach Lacrosse, and he is excellent. This guy, he got an A in cell bio when he took it with me. Really bright, med school bound. And he also has access to the exams. All right. So he's not going to give you the exam questions, but he knows what's on the test. Right. So he's going to be able to help you out, you know, in a similar way that I am, you know, knowing what sort of information you need to know. And he's just really, really good. You guys are going to need, you know, there's a lot of students in here that will need the tutoring. There's some students that will use it, and there will be a dramatic difference in grade between those students that use it and those students that don't. Okay. He's really great to work with too. Very friendly guy if you don't know him already. But so you also have me as a resource as well. Okay, for extra help. The thing that you can get from him though is very long periods of extra help. Now I think he's going to probably put together a video recording and just kind of introduce you to the center, the tutoring center, and how to make an appointment and things like that. Okay. All right. Okay, so we got quizzes, we got exams, and then we also have these data discussions, right? So I really think that at this level in your career, trying to read a scientific report or scientific paper, it poses there's a lot of barriers to that, right? They're extremely difficult at this level to really grasp and get through. Very extremely time consuming to get through something like that. An important first step in being able to read and understand scientific reports is being able to understand how the data is being presented to you. And that's what we do with these data discussion sets. So basically what I'm going to do is give you some sample data from a cell or molecular biology journal or study. And then there's a few questions about it, and I want you to answer the questions. Simple, simple. However, I do want to go over an example. And this example will kind of explain what I'm looking for in your responses. You know, ChatGPT, you could put these questions in there and upload the figure and you'll get an answer. I'm not prohibiting you from using ChatGPT to assist you in answering these questions though, or Google or whatever. There are, in most of these data sets, there are things that you're going to need to look up. You're not going to know. I just want to kind of go through this specifically as an example, though. So let's look at this image here. And, we haven't had any background information, and that's okay. All right. But what this is a, well, it tells you what here. So you're looking at mast cells and how they're synthesized. They store histamine in the form of these membrane-bound granules. Upon stimulation of these cells, histamine is released and triggers allergic reactions in tissues. The following figure shows the result of an experiment in which the mechanism of histamine release was studied, right? So electron microscopy image, and then it's got all of these, things within the figure labeled A through C. A suspension of mast cells treated with a drug that induces histamine release. After 10 seconds, the cells were quickly fixed in cold formaldehyde solution and processed for electron microscope analysis. Sections were stained with uranyl acetate and lead citrate. Now, in our lecture, I kind of breezed through. I'm going to tell you right now, like, you're not going to have this question as a question to answer, but I breezed through the electron microscopy stuff in the book a little bit. We didn't cover any of the stains or anything that are used in that. So unless you remember from whenever you learned it, which probably was never, if you're a typical biology major, what uranyl acetate and lead citrate do, that's something you're going to need to look up. And that's fine. I want you to be able to look it up. If you don't remember what mast cells are, look it up. Understand what's being presented to you and understand the questions. But at that point, what I want to see is how you develop the answer. So this is a student's response from a few semesters ago. And he actually doesn't get, he misses the mark here on a couple of them. But I gave him perfect credit or full credit because of the way that he answered the question. And that's what I'm looking for. I'm looking for you to kind of prove to me that you're working through the problem. All right. So let's look at what he wrote here. And it's almost like he's speaking out loud, which is a really good way, in my opinion, to approach this. As you're thinking about the problem, write what you're thinking. So he says, well, this is funny. When I first look at the picture, I thought I was looking at many cells. That makes sense. I could see someone looking at this and be like, oh, look at all these cells here, and they're blowing up. But you think about it a little longer, and what we learned, electron microscopy is, it's really, really zoomed in. This is just a section of a cell, right? These are all vesicles that are being produced and releasing histamine. So I realized I was looking at an electron microscopy image. So we're just looking at a portion of the cell. And okay, these are mast cells. All right, in chapter 11, since this question is from chapter 11, and each of these data set questions are going to be associated with the chapter, and that should help you kind of, narrow down what sort of topics the data set question is getting you into. We learned about how vesicles form part of the endomembrane system from the endoplasmic reticulum. So I think A is either nuclear membrane, or it could be chromatin, or it could be both. All right. Now, the answer is heterochromatin, and he didn't give you that. And that's okay, because what he's showing me is that, and I keep saying he, and I don't know if this is from a he or a she. I apologize for that. But what this student is showing me is how they're deducing the answer, right? How they're using their background knowledge to apply it to a study and start to develop the answer. When you answer these, I would like you to do that in that format. You're going to have a hard time, just for those ChatGPT lovers, having ChatGPT write out a script like this. That's going to, not pass my eye there. Okay. So do it like that. This is a really important component to your development as a scientist, being able to interpret data sets. These data sets are, again, they're really small portion of your grade. They're there to boost your grade. All right. I'm not going to be grading these difficult. What I'm really going to be looking for is that you're just attempting it, right? So, basically write out loud. Tell me what you're thinking, and, use the book, go into the book, try to get some ideas. And, that's it. If you take this class in person, this is something you get to do with other students in a group. I haven't really developed it in this course to be able to do that. Okay. So for every unit, you have one of these. They do have due dates. All right. Yeah. So, and it's a different date from the exam. So there are, but it's the same date as the quiz or close to it. All right. So this course, you have a lot of due dates in here, and I'm doing that on purpose. Sorry, because I want you to stay up on the material. You can't wait for the night before to study for these exams. Okay. You really have to build yourself in this course with this sort of knowledge. All right. How to exceed on the exams? Study, study, study. Read the book, understand the book, understand what I'm talking about in lecture. Work the problems at the end of the chapter. All right. That textbook has the answers to the problems at the back of the book, so you can reference that. But don't just go to the answers. All right. Attempt it. Give it a chance. If after 5 minutes you just have no idea, then go to the, check the answer in the back of the book. Okay. We're going to cover pretty much the entire textbook. There are a few sections that I have removed because I really think that they are, well, they're just not the most important sections in the book, but we do cover about 90% of this textbook. So we're going to cover a lot of information. We're going to cover it quickly, and we're going to study it in depth. All right. Taking this course is going to really put you in a position to succeed in molecular biology if you need to take that course as well. And, I really would like you to give this material a chance. In my opinion, things that are boring and difficult, right? Or boring or difficult, trying to explain this stuff, but it's easy to muddle the two, right? Once you start to understand these concepts, I think my hope, and we're all biology majors here, and my hope is that you will start to really appreciate it, especially the complexity of it. Okay. All right. I don't want to scare you guys at all. We're going to have an in-person relationship. I'll see you guys on Thursday for lab, but I just want you to know, the level, the type of work that I'm expecting from you guys in this course. And I know you can do it. So, and I'm here to help you succeed in any way possible. Okay. Again, really looking forward to meeting most of you on Thursday. If someone is not enrolled in the lab, that is okay. I know that they're co-requisites, but maybe someone, I don't know. I haven't really checked to see if every student who's taken the lecture is taking the lab. I should probably do that. But that's it. So welcome back to school. I hope everyone had a wonderful break. And that is it. So I will see most of you guys on Thursday. And, best of luck in this course. Work hard, and it's very manageable. You'll do fine. All right. And that is all I got.